Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 11th biology students, and this is for 29th August 2020. Our memory helps us in tracing out what we have been studying in the chapter breathing and exchange of gases. In our last class we were talking about various disorders associated to respiratory system. And to conclude this chapter, we are going to talk about an interesting curve called oxygen hemoglobin dissociation curve. What is this oxygen hemoglobin? The relationship between partial pressure of oxygen, PO2, and percentage saturation of the hemoglobin with oxygen is graphically illustrated by a curve called oxygen hemoglobin dissociation curve or oxygen dissociation curve. So how exactly this happens? So basically you can see oxygen carrying capacity. So transport of gases will take place, and the blood is the medium of transport of both oxygen and carbon dioxide. And particularly oxygen, as you are seeing, oxygen is carried by blood in two forms. Dissolved gas in plasma about 3%, and as oxyhemoglobin by RBCs, as you are seeing, 97%. So the oxyhemoglobin dissociation curve is an important tool for understanding how our blood carries the oxygen from the alveoli and release the oxygen to the tissues. So you can see your blood entering into alveoli and going to the tissues. So the hemoglobin is a pigment, I said, if you recollect from my memory. It has got liking for oxygen. So oxygen plus hemoglobin forms oxyhemoglobin, and this hemoglobin is present in the RBCs. And the hemoglobin molecule, as such, you can see heme. So it is in red blood cells, contains several 100,000 hemoglobin molecules. And this is what we were talking some time back, how the exchange takes place. And for that I said three reasons. One is the partial pressure of carbon dioxide, another is partial pressure of oxygen, and third is anatomy of the alveoli of the lungs. So finally the oxygen reaches the tissue cells and carbon dioxide goes out of our body. To understand what is the purpose of this, to understand respiratory function, it is extremely important to know how oxygen gets attached to hemoglobin in blood within lungs and how it detaches from hemoglobin in the tissues. To show the equilibrium of oxyhemoglobin and non-bonded hemoglobin at various partial pressures. You can see hemoglobin carries oxygen and carbon dioxide as well. So inside the red blood cell, and once carbon monoxide binds very lightly to hemoglobin, what happens? Oxygen and carbon dioxide can no longer be carried. So they are to be out. Now this is how we represent the oxygen hemoglobin dissociation curve. It is in the form of S shape, so we say sigmoid curve. A sigmoid curve is obtained when percentage saturation, you can see on the left side, on y-axis percentage of saturation of hemoglobin, with x-axis partial pressure of oxygen is plotted against the PO2. The partial pressure of oxygen of the air within alveoli is approximately 105 mmHg. The partial pressure of oxygen of the blood leaving the alveoli is about 100 mmHg. This is because the blood plasma is not completely saturated with oxygen. So we have got the percentage of hemoglobin that is bound with oxygen is called percentage saturation of hemoglobin. At this low partial pressure of oxygen, the percentage saturation of hemoglobin accounts for only 75%. Okay, you can see it. And the oxygen hemoglobin dissociation curve is shifted either to left or to right by various factors. Partial pressure of carbon dioxide, H plus ion concentration, temperature are some of the factors in shifting this. So shift to right, we can see oxygen hemoglobin curve is a sigmoid shaped curve. It represents the relationship between concentration, that is oxygen concentration and percentage saturation of hemoglobin. This curve describes the relationship between available oxygen and amount of oxygen carried in the hemoglobin. In this, what is important? Partial pressure of oxygen is on x-axis and oxygen saturation is on y-axis. So the horizontal axis is the pH02 or the amount of oxygen available. The vertical axis is SH02, that is the amount of hemoglobin saturated, that is saturated oxygen with oxygen. Once the PaO2 reaches 60 mmHg, the curve is almost flat. So PaO2 of 60 or more is usually considered adequate. But at less than 60 mmHg, the curve is very steep and small changes in the PaO2, that is the amount of oxygen available, greatly reduce the SH02. Traditionally, the curve starts with normal, that sigmoid curve is pH should be 7.4, temperature should be 37 degrees centigrade, and PaCO2 partial pressure at 40, right? These are the three factors. And anytime when there is some changes, then we say shifts. Changes from these values are called shifts. The term affinity is used to describe oxygen's attraction to hemoglobin binding sites. So affinity changes with variation in pH, like normal is 7.4, any variation in the pH it changes. Temperature is 37 degrees, change in temperature changes. Partial pressure of CO2, and there is a substance 2,3-DPG. So when the curve shifts to left, okay, hemoglobin has increased affinity for oxygen and releases less to the tissues. So you can see the curve is shifting to the left. The oxygen hemoglobin dissociation curve, when it is shifted to left, then the following conditions must adhere to. What is that? That is the fetal blood, because fetal hemoglobin has more affinity for oxygen than adult hemoglobin. Decrease in hydrogen ion concentration and increase in pH and decrease in partial pressure of carbon dioxide. These are the conditions. Then the curve, as you are seeing, will shift to left. And then when it shifts to right, when the curve shifts to right, as you are seeing, right shifted, hemoglobin has a decreased affinity for oxygen. The oxygen hemoglobin dissociation curve, when it shifted to right, the following conditions must be there. Decrease in partial pressure of oxygen, increase in partial pressure of carbon dioxide, increase in hydrogen ion concentration, and decrease in pH, and increased body temperature, and excess of 2,3-DPG, diphosphoglycerate, a byproduct, remember, a byproduct of glycolysis. Now, when these conditions are there, the curve shifts to right. So left shift shows an increased hemoglobin affinity to oxygen. It is easier to load oxygen but difficult to unload it. When the curve shifts to right, it shows decreased hemoglobin affinity to oxygen. It is difficult to load the oxygen but easy to unload the oxygen. And factors and how the curve is affected, that we just now we discussed. Variation of the hydrogen ion concentration, effects of carbon dioxide, effects of 2,3-diphosphoglycerate, temperature, carbon monoxide, effects of METHEMOGLOBINEMIA and fetal hemoglobin, and the variations of the hydrogen ion concentration. This changes the blood's pH. H plus ion. What is actually pH? It is the negative logarithm of H plus ion concentration. So when there is a change in H plus ion concentration, naturally pH changes. A decrease in pH shifts the standard curve to the right, while an increasing hydrogen will shift to left. This is known as Bohr effect. And then effects of carbon dioxide. Carbon dioxide affects the curve in two ways. First, it influences intracellular pH, Bohr's effect. Second, CO2 accumulation causes carbon compounds to be generated through chemical interactions. So low level of carbamino compound have the effect of shifting the curve to the left, while higher levels causes rightward shift. And what about effects of 2,3-DPG? 2,3-diphosphoglycerate are created in erythrocytes during glycolysis, as we discussed in Respiration chapter. The production increases several conditions in the presence of diminished peripheral tissue availability such as hypoxemia, chronic lung diseases, anemia, and congestive heart failure. High levels of 2,3-DPG shift the curve to the right, while low levels of 2,3-DPG cause a left shift. And this is seen in septic shock and hypophosphatemia. And the temperature does not have so dramatic effect as the previous factors, but hyperthermia causes rightward shift while hypothermia causes leftward shift. And effects of METHEMOGLOBINEMIA, it is the form of abnormal hemoglobin and it causes leftward shift in the curve. And methemoglobin results from the change of the ferrous Fe2 plus to Fe3 plus. The Fe3 plus does not effectively bind oxygen. And carbon monoxide also has the effect of shifting the curve to the left. With an increased level of carbon monoxide, person can suffer from severe hypoxemia while maintaining normal partial flow of oxygen. And what about in babies? Fetal hemoglobin is structurally different from normal hemoglobin. The fetal dissociation curve is shifted towards the left as you are seeing, and it is low fetal arterial oxygen pressures. So thus the oxygen hemoglobin dissociation curve indicates whether how much affinity is there for the hemoglobin to the oxygen, and this helps us to understand the loading and unloading of oxygen into the tissues. So this is a graphical representation. What you have to do is just plot the graphs, try to understand whether the shifting is towards left or right. If the graph shifts to left, what are the conditions that make the graph to shift to left, and what are the conditions that make it to shift to right, and what happens if the graphical representation is towards the right or towards the left, how it affects us in making oxygen available to tissues. So basically what we are talking about is it is the transport of gases